Hello, good morning, good afternoon, good evening to all of you joining from different parts of the world. Please let us know where you're joining from. Write that on the chat. Today we have a special guest speaker, Dr. Ram Nair from the World Vegetable Center. Ram has a very extensive trajectory in plant breeding. He graduated from the Kerala Agricultural University in India with a PhD, and then he worked several years in Australia in forage breeding before joining the World Vegetable Center as a Mungbean breeder, or more specifically vegetable legume breeder, because his main work is in mung beans, but he also works in other types of vegetable beans. Dr. Nair, it's a pleasure to have you here. I have enjoyed reviewing your program, although I didn't visit you personally last year. I wish I could do that soon. So I'm gonna give you the floor right now, and I'm gonna stop sharing so you can start sharing, okay?
Thank you, Carlos, for the kind introduction. Morning, everyone.
Yes.
Good
You're sharing
afternoon.
very well. Go ahead.
Good afternoon and good evening to those who are joining from different parts of the world. It's a great opportunity and a privilege to talk about learning from breeding mungbeans within an international network. For those who are not familiar with mungbean is Vigna radiata, subspecies radiata is one of the most important warm season legume grown mostly in Asia. It is, it's a significant amount of protein, carbohydrates, and a range of micronutrients like iron, zinc, and also good source of folate. Currently, mungbean is grown in about 8 million hectares worldwide, mostly by smallholder farmers. As I said, mostly in Asia, but it is rapidly spreading to other parts of the world. The average yield or productivity is around 720 kilograms per hectare, but the potential yield is around 2.5 tons per hectare. So you can see that the big yield gap. Now, to make sense of how the crop fits into cropping systems, this is an example from the Indo-Gangetic plains. So rice is grown during the rainy season, the monsoon season, so southwest monsoon season starting from June, July, and then it's followed by wheat in the cool season. And between the wheat and the rice crop, there is a gap of about 80 to 90 days, and mungbean, the short duration varieties, which we now have even 55-day varieties, and they fit in well in this fallow system. Now, of course, being a legume crop, and it's add to the system of having a continuous cereal cropping, so adding to the nitrogen to the system, of course, also being a break crop. Also, mungbean is grown as well as a monocrop, as well as an intercrop. So here I give examples from Kenya, where mungbean is grown as a monocrop, as an intercrop, pigeon pea here as an intercrop with maize. And there are instances where mungbean is grown as an intercrop in sugarcane also. So there are different ways in which the mungbean crop is grown. And already, Carlos, in the introduction, there were good photos of different mungbean products. So again, just to show a case for those who are not familiar, Mungbean whole grains. Then you have got them, you may be familiar with dal if you have visited any Indian restaurants. So moong dal or dal is split grains. Then you have got the sprouts, mungbean sprouts, which is very popular in Southeast Asia. Then you have got mungbean starch, which is very much sought after for noodles. Then mungbean milk. Then the seed coat of mungbean is used to make a health drink. Because the seed coat has got chemicals like vitexin and isovitexin, which helps to reduce body heat. So that has been used to develop a health drink. So different products have been developed, and as a weaning food, so many different products have been developed, which are all healthy. So coming to the International Mungbean Improvement Network. So the International Mungbean Improvement Network was started in 2016, funded by the Australian Centre for International Agriculture Research, ACIR. And now we are in the second phase. And we have partners from Africa, from East Africa, KALRO, Kenya Agriculture Livestock Research Organization, from India, the Indian Institute of Pulse Research, from Bangladesh, the Bari Agriculture Research Institute, from Myanmar Department of Agriculture Research, from Indonesia, the Indonesian Legumes and Standard Testing Institute, from Australia, we have the Department of Food and Agriculture, Queensland Government, as well as the University of Queensland. And this network is led by the World Vegetable Center. We have got also support from the Syngenta Foundation for Sustainable Agriculture. So when we work with the partners from different countries, it's very important to have product profiles developed for different countries. And so here, if you look for the product profiles, you can see the must-have traits, which is in dark green. So if you see India, Indonesia, Myanmar, and Bangladesh, you can see MYMD. This is Mungbean Yellow Mosaic Disease, which is the most important disease which is responsible for the lower productivity. And so that is why this is #1 priority. We have got excellent varieties now, but still we are trying to get the more adoption of these resistant varieties so that we can get the productivity, we can increase the productivity further. But when you look at the product profile of a country like Kenya, you can see that there is no, when we took samples, there were no bigomoviruses. Mungbean yellow mosaic is a bigomovirus, but we found that there is the bean common mosaic virus and some instances cucumber mosaic virus. So we have the impact pathway, which is very important. And so if I quickly go through it, we have the Mungbean collection, then we have identified key traits. I will, in the next few slides, I will talk about the importance of this collection. At the World Vegetable Center, we have over 8,000 accessions of Mungbean, from which we have developed a core collection and then a mini core collection. We have identified key traits, and then markers have been developed or are being developed for both biotic and abiotic stresses. They have been introgressed into the elite lines, and the lines developed have been tested at multi-locations. And then there is, in some, I have got examples where variety releases have happened and farmers adopting them. And in parallel, as part of the network, we do capacity development, national breeding programs in terms of breeding methods, digital data collection, developing product profiles. And these product profiles are developed in consultation with the major stakeholders. It's not the breeder sitting and developing, it's in discussion with the major stakeholders. And then we have a very important component is strengthening the seed delivery systems. and being a legume crop, very few companies, private companies, are interested in promoting the seeds. And so this is a big area, or a big area where we have to work further, and I will talk about it in the next few minutes. And development outcomes, we are looking for farmers having better, higher incomes, improved human nutrition, and very important also from a soil point, and also for sustainability, enhancing the soil fertility. So I mentioned earlier the partners, specifically I mentioned about the organizations. So that is, those are the funded partners, but the network is beyond, more than a project. It's a program in which, in a larger network, where organizations, we are already signed with 20 organizations. And so this is, so we have extended our activities, especially the sharing of germplasm, and also the germplasm accessions as well as improved lines have been done with other partners. So I mentioned about this mini core collection. So from the 8,000 accessions, we had developed a core collection by phenotypic characterization, and then that's about 1,500. And from that, we developed a mini core of 300 accessions by molecular characterization. And this has been shared with different partners. And then our molecular biology team, in collaboration with the National Taiwan University as well as with the Vavilov Institute, did this DART sequence analysis of the mini core in comparisons with the crystal. It's a very important variety in Australia, and BC 1973, which is the variety which has been used for sequencing. And then you can, as expected, the mini core is all over the place. And then you can see the accessions from the Central Asia concentrated here, and there is the South Asia here. So there is the mini core, as you expect, is everywhere. So we also did resequencing of these Mungbean accessions by Illumina, and thanks for Roland Schaffleitner and Dr. Churin Riley for getting the grant for the Illumina sequencing, and we have done that, and SNPs have been developed. And very important is that the mini core collection, which has been developed, the 300 odd accessions, to be specific, it is 293 accessions, which have been shared with different partners. Here we show that this has been shared from Asia and in sub-Saharan Africa. A lot of data, still analysis is in progress, and we're now in a couple of traits we have already done. So still analysis is in progress with the data sets we have obtained from all these sites. So talking about data as part of the Mungbean network, when we started, the partners were collecting data by using feed books, but then we started implementing this KD-DART database management, which is a plant breeding database management supported from diversity array technology from Australia. And here, one of the major attraction of the partners and from my own perspective was that some of the varieties are, at least there is one variety which is known by 10 different names in different countries. So it is very important to have the pedigree of these varieties all, put in one place so that breeders can know what variety they are using, so that they don't cross the same one and make mistakes. So the pedigree is there, data collection is there. it has not been. So the topic of this seminar is on learnings. One of the learnings it's not that easy to implement database management, especially when there is staff turnover with the partners. But we do it continuously so that there is a recognition of this. And then, but some staff member, some organizations have taken it as a very core one and started implementing, and so that at least 2 staff know about this, and they are also training their own colleagues afterwards. So this is very important, especially now with this when we are getting all these phenotypic as well as genotypic data and getting them used in the breeding program. So we can have this collection by using the phones. Now earlier it was Android, now iPhones are also being used, so very good. And so I think the partners appreciate it, and so that is something which still we are working on it so that it's implemented more successfully. But what we have learned is to make sure that there is continuous training program done, and there are fine-tuning of these as is common in each different software database softwares. And so, but we get very good support from the diversity array technology from Canberra in Australia. Now I will quickly go through the different traits. So if you see the picture, so mungbean is also known as green gram, but this is a shiny green gram, dull green seeded, but you can get yellow seeded ones also. But there are black seeded ones also, but these are the major types. So there have been GWAS has been mapping done, SNPs have been identified. And then we have using the mini core collection, GWAS analysis has been done. And we have extra early and medium varieties. Canopy traits, we have in collaboration with ICRISAT, we have done the canopy analysis and identified lines by using LISI scan with our colleague Kritika and Bhajan from ICRISAT. And then also in our headquarters, we have looked at the correlation between the green pod biomass with the different parameters of reflectance. And also when not often the roots parameters are looked at, but in collaboration with the Indian Agriculture Research Institute, we have looked at the variation for the root length of the mini core collection. There is good significant differences. And also we have studied the phosphorus use efficiency and found the SNPs in different, in this case, chromosomes 8, 4, and 7. Earlier I mentioned about the mechanical harvesting, which is a trait, an agronomic practices which is on the increase, because traditionally mungbean is hand harvested, but it's in countries like Australia, it is 100% mechanically harvested. In other countries, because of labor issues, this trait is, this mechanical harvesting is on the increase. And when we look at screening of the mini core, we found a couple of accessions which have showed leaf senescence, natural leaf senescence at pod maturity. So for those who implement the mechanical harvesting, you may be familiar that you have to use a desiccant like glyphosate before you can mechanically harvest. Otherwise, the produce will not have the good quality because of the crushing of the green saps, especially if the plants have not turned fully dry. And so some varieties still have the greenishness, but the leaves are still green at pod maturity. That makes it not acceptable of grain quality, because spotting will happen on the grain. So This trait is under investigation. It shows that it is preliminary studies have shown that it is a heritable one. So still we are now studying the mechanism. Now coming to the biotic stresses, I mentioned about Mungbean yellow mosaic disease. This is the most important disease. Some of the very great work done by Worldwide in collaboration with the National Institute of Nuclear Institute of Agriculture Biology in Pakistan, and with our partners from Keshatsat University, Mungbean NM94 is the mutant line a variety was developed through mutagenesis, and that has been several varieties. So that particular line has been released as in different names in different countries, very popular. But then 10 years back, we found that that particular line or this was becoming susceptible. It's because of a virulent strain of the virus, particularly in the northern part of India. And in collaboration with the Punjab Agriculture University, we identified lines ML1628, ML818, and with that, we have now developed good improved lines with very strong level of resistance to the yellow mosaic disease. And that has been, those improved lines have been shared with our partners. And very recently, now there was an International Mungbean Congress, which I will speak towards the end of my talk, that there was a talk by Yaping Ning on the QTLs developed for yellow mosaic disease. So this is a welcome thing, and which is very critical for our partners to implement in the breeding program. And we have got traits like powdery mildew. Again, here I forgot to mention that that in sub-Saharan Africa, it's the bean common mosaic virus, which is prevalent. And so we have looked at the mini core, and one pleasant thing is with the mini core collection, we are getting pleasant surprises each time when we evaluate them. So yellow mosaic disease, very good variation. Then because we were expecting all the accessions to be susceptible, but it was a surprise that we found at least 5 which were holding on well in this India, Bangladesh, and Myanmar. So that's again additional sources of resistance to the disease. Then powdery mildew, again the mini core collection, good variation seen. And in the case of anthracnose, the fungal disease, another fungal disease. So both these, for both powdery mildew and anthracnose, we have identified good lines, good accessions with good level of resistance to both these diseases, which is we have now started using in the breeding program. Dry root rot, Macrophermina faciolina, is a disease which is emerging, especially where Mungbean is grown as part of the rice-based systems. And we have, again, the mini core give us some good lines, good accessions, which is, you can see those, some accessions in the paper towel method as well as in the pot assay. Coming to bacterial diseases, tan spot and halo blight, these are very important in Australia. Fortunately, it is not an issue in the Asia at the moment, but it could. So they are seed-borne, so very we have to be extremely careful. And again, with the mini core, we have identified accessions. Yes, in this case, one accession for tan spot and this particular accession for halo blight. So you can see that halo blight, how devastating it can be. And it's at the moment a very high priority for our Australian collaborator. Coming to insect pests, Brookits storage insect pests, we had some good work done in the past. We had earlier, it was the sublobata, TC 1966. Then we had 2802 and 2709. These are Igna radiata accessions identified in the past, and that which we have developed markers And this is the first trait in which we have used for marker selection. And we have also looked at the cooking duration of the brookit resistant lines, and there was no significant difference compared to the commercial checks. Thrips can be devastating or damaging, especially during the spring season when the temperature goes up in the Indian subcontinent. So in India, Pakistan, Bangladesh, this is a major issue in the spring sown crop, and it can be confused with heat stress also. But you have to look carefully. You can see this. It can, especially if it is in the flowering stage, but then the thrips can attack also during the seedling stage. Then cowpea aphids are also a problem, and but it could be managed. But again, in the mini core, we have identified resistant lines or tolerant lines. Now, moving to abiotic stress, heat tolerance. Mungbean is a drought hardy crop, but still, when you are extending the cultivation of the crop to, for example, in Central Asia, in Tajikistan, the temperatures went up to 45 degrees Celsius. But some of the lines which we have identified through collaboration with the Chandigarh, Punjab University, Chandigarh, and in India, and we had identified heat tolerant lines, and some of these lines are doing very well under 45 degrees Celsius. And but please bear in mind, these are under irrigated conditions, but still the temperature, ambient temperature is very high. And so Mungbean is able to grow under such conditions, provided there is sufficient water in the soil. And it's very likely that a variety will be released in Tajikistan very soon. And when you extend the cultivation of the crop to other areas, for example, in countries like Bangladesh, waterlogging tolerance is important, as well as salt tolerance. Sometimes they come together. And again, the mini core collections were evaluated. This is work done by one colleague from Myanmar, studying from the University of Western Australia, that identified SNPs and from using the mini core collection. And one Breria worked in our headquarters looking at salt tolerance at a seedling stage, and SNPs were identified. So SNPs or QTLs have been identified, and so now they are in the process of being utilized in our breeding program. And still continuing with the drought adaptation, this is the collaborative work with the National Institute of Abiotic Stress Management in India. We have done LamnaTech facility using, comparing Mungbean genotypes with water and under stress conditions, and predicting the biomass to compute the plant growth rates as well as the water use indices, and so what sort of response these genotypes make in response to the soil moisture stress. We also, in collaboration with ICRISAT, Lycée field, we have drought imposition at reproductive stage, and looking at identifying types. So interesting genotypes with high seed yield, high transpiration efficiency, and low water demand. So these are some of the lines which have been identified. So we are using different techniques and in collaboration with different partners in identifying lines which could be used in the breeding program. And nutrition is important. Mungbean is a very good source of nutrition, but further it could be improved. And we had identified lines which could have a higher uptake of iron when grown in soils with more high available iron. And those lines have been crossed with the elite lines, and the improved lines were shared with our partners as part of a Africa Union project in Kenya, Tanzania, and Uganda. And if everything goes well, at least one variety from all these three countries will be released this year. And the protein, so this is the buzzword now for plant-based proteins, and Mungbean is well sought after, especially because of its unique protein nature. And we have, in the Minico, we have seen good variation, and now we are looking at how best, so using Nair techniques and for calibration of the prediction of the protein content. Earlier we had used wet chemistry, but now this we are using the Nair for prediction. In collaboration with the partner in the University of Queensland, we have trying to fix the other areas where the network in the first phase did not, because the University of Queensland came to the joined the network in the second phase. So very good skills coming into the network with artificial intelligence guided breeding, speed breeding in some countries, and then helping us in the using for the QTL identification, and some preliminary work in gene editing is happening. One of the PhD students, Shanice Van Haften, is working on the drone-derived traits to look at the canopy growth in the Mungbean Minico. Again, good variation obtained or realized. There is another student, Kathleen Dudley. She is looking at the Mungbean flowering behavior on as a guide for crop improvement. So with the aim to use them in the as key targets for the in future for gene editing pipelines. So whatever techniques you may use, it is very important that you have the evaluation of lines done in a participatory manner. So involving the major stakeholders, especially the farmers, and so these are photos I'm showcasing from our collaborators, Karmi from Kenya, Papias Bingo from Tanzania, and Emmanuel from Uganda. So very good interaction is there, and there is a lot of involvement, and they go and score these each and every line for the different traits, seed size, seed color, then maturity, disease resistance, and some cases they even cook them and see whether how they are good in terms of cooking time, and that sometimes Mungbean, some older varieties, they were stony, so they are very cautious in recommending. So making sure that the improved lines are very good in terms of cooking quality. So it is very critical that we involve the farmers during the varietal testing. And so I am happy to say that the varieties in Kenya released in 2017, Karambo, Baishara, and Nedugo Tosha. These are all large size, because traditionally when I visited in 2011, they were growing varieties which were small seeded and also particularly late maturing, about 90 to 95 days. And then when we introduced these early maturing varieties, there's a big difference. So within 2 months, 2.5 months, they could have a crop, and wherein especially where there was problem in getting good soil moisture. And the adoption of this is very good, and thanks to great work done by our collaborator Carmeria. And in Tanzania, 2 varieties were released in 2022, Tari Green Gram 1, Tari Green Gram 2. And both these varieties, again, because of their earliness, well accepted. And you can see that the color is brown color. This is very much sought after in Tanzania, especially in the domestic market. So very interesting. So in 2002, as in 16, Mungbean variety released in Myanmar by the Department of Agricultural Research. It has got shiny bold seeds. It is resistant to both yellow mosaic disease as well as dry root rot disease. And the most important spectacular thing is that it has got better sprouting quality. And this is it's very important because Myanmar, the mungbean crop is grown as a cash crop. They take great care for the crop. And there is companies like Ever Special, which is the Europe's largest sprouting company. They procure mungbeans from Myanmar and China. And we have a project implemented, funded by DENIDA, wherein we are trying to fine-tune, making sure that good agriculture practices are followed, so that especially in the sprout market segment, which is a premium, very choosy, very particular with regard to chemicals, because when you're consuming sprouts, you have to be extremely careful. And that in that way we have implemented good agriculture practices, so that there is no residue issue of chemical residues in these products, which are being used for export. But we are also encouraging the community in Myanmar to consume, also as part of improvement of the nutrition. Talking about varieties, earlier I mentioned in the impact pathway, the critical one is because of the less support from the private sector, we have to work very hard to get the mungbean varieties to the farmers, the research institute, the universities, the non-governmental organizations, and some private companies. In this instance, I'd like to acknowledge the Syngenta Foundation for Sustainable Agriculture, which has been helping us to link with some of the non-governmental organizations and our partners in different countries. For example, the Nair in Uganda, they are piloting community seed schemes, wherein they produce quality declared seed, and then which is made available to farmers. So different methods are, so formal and informal systems are there, but very critical that the improved varieties reach the farmers on time. And because we talk about speed breeding, but we need to make sure that once we develop good lines, they reach the farmers on time. And when you say on time, is before the season starts. And I have to talk about nutritional point. And when we were in the African Union project, which from the Mungbean network, we developed improved lines. For example, the improved iron content lines. They were tested, and also since in Uganda the crop crop was not popular, we had to develop recipes, and our collaborator Emmanuel and his team did a great job in coming up with all these different recipes. And so recipes which are in Asia could not be used directly, so they have been customized. So different very good dishes for Uganda, and some they have taken from Kenya. So lots of beautiful dishes. I had the privilege to attend the field day, and wherein these recipes were showcased. Again, talking about value-added products, there is a company called Nature Lock, which is promoting Tuesdays. This is ready-to-eat mungbean product by using natural ingredients. And one of the varieties, if you remember, we had Beheshara, Nedugu Tissue, and Karimbu. But of the three varieties, Nedugu Tissue, which was chosen and for its suitability for industrial pre-cooking. And now they are linking with Farmer Network through Calro. And this is Ms. T. Mukunyaunda, who's a co-founder, executive director, who explained to me the process, and they are now supplying these Tuesdays ready to eat to the children. And I think this will develop further, and large volumes will be needed. And this is sort of the things which we need in mung bean to make sure the value chain is strengthened so that the farmers, they get good income and there is steady market for the products. And very critical is that the products which are developed are not having additives or preservatives, but developed through natural ingredients and which will help to solve the malnutrition. The network, we have released Mung Central, which is a newsletter, which is, I encourage each and every one of you to read. We have so far released 10 editions. Each edition has got a recipe. So those who are interested in mung bean, please go to this website, download the recipes and try it. I'm sure at least you will like at least a couple of them. And we had also some publications have come out with this collaboration, this network, this is the Mung Bean Genome Work. And I must say that the Mung Bean Network has been a platform for developing other projects. I mentioned about the Africa Union project. There are other projects like the Danida project, which like they wanted sprouting. So we had worked in the network. So the products developed from the Mung Bean Network is feeding into these developmental projects. And also we were fortunate to have an Asia-funded harvesting project, which helped to optimize the harvesting methods in Bangladesh, Myanmar, and Pakistan. And so that, again, we have to make sure all the agronomic practices and also the cultivation practice, agronomic as well as the harvesting and the post-harvest practices all are adopted in well, and also they are all strengthened so that at the end of the day, you get a good produce. And very important that the farmers, they make a good life, have a better livelihood by growing mung beans. So very happy to see that the recent WACS or the Vision for Adapted Crops and Soils released by the last year by the US Department of State, which in collaboration with the African Union and the Food and Agriculture Organization is supporting scaling up of and also research in selected crops. Mung bean is one of the crops selected for scaling up varieties for better nutrition and livelihoods. And we are now working with CIMMYT and our partners in scaling up of the varieties, which is a very critical step to get these improved varieties adopted by farmers. So this is a timely intervention by the US Department of State. And last month, we had the International Mung Bean Congress, wherein we had 100 participants from 25 countries. Very good interactions, very happy to get all these participants from different countries in one forum. And I acknowledge the donors and partners. And it is all teamwork. I don't want to name each person, but it is the teamwork which has helped us to tackle these issues. So each country is unique, and we need to, and we have to make sure that we interact with the key stakeholders so that we learn from them, and then we go back and then make sure the products we develop are adopted by farmers. And when you do it in a participatory manner, the adoption can be, increased tremendously. Thank you for your attention.
Thank you, Ram. What a wonderful summary of what can be done in a crop that doesn't have the support as other crops have. So you summarized it very well, and I appreciate that. I would like to read some of the questions, which we have many already in the chat. It says, regarding insect resistant to bruchets, the seed color is varying from shiny green to brown and dark. Is it due to genetic mixture or related to maturity of seeds?
Yeah, so in terms of bruchet resistance, the color is not important. We have identified bruchet resistant lines which are green and shiny, a dull green, And so there is it is genetic resistance. So we have got two sources of resistance, V2802, V2709. They have been used, and we have got good levels of resistance. So this is resistance to Calosobruchus chinensis and Calosobruchus maculatus. But then since you asked about it, I thought I will little bit explain more. So, but when these resistant lines were screened in Kenya, we found susceptibility. So we are now looking at more accessions, especially the mini core, and one of the accession is showing good promise. But then one of the brookid species, Calosobruchus analis, is on the increase. So none of the mini core accessions were found to be resistant. But fortunately, when we looked at our related species, sublobata, a couple of sublobata accessions have shown resistance. So it is an important issue, and we are trying to have a good level of resistance. Thank you.
A related question is whether the seed luster, the shininess, has anything to do with disease resistance.
No. Earlier we thought that it is due to that, but that is why I said we have got now brookid resistant lines which are green shiny and dull green.
Okay. A question here is: are you working on F1 hybrids for Mungbeans, or is all pure line development?
Yes, so we are at the moment all are open pollinated varieties. There have been some interest in having hybrid Mungbeans, but so far, some progress made by having an open flower type by the Kesetsat University. So there is. But again, we already have challenges in seed production as an open pollinated variety. So, getting a hybrid, I see the pigeon pea hybrid program in ICRISAT. There are challenges, but I think still we will keep working. But if we need to have, we have to make sure that it works in a commercial way. So, yeah. But we still keep investigating. Yeah.
Okay. Yeah, the grain color and cooking, does it stay the same color as the grain, or does it change, or it depends on the variety? Well, after you cook the Mungbeans. And what is the typical cooking time? That's part of the question too.
Yes. So the the color remains intact. So because that is why there is a lot of preference for the color. And so if, for example, in Indonesia, there is preference for dull green. In the Indian subcontinent, there is preference for green shiny grains.
Okay. And the cooking time, approximately?
Cooking time, you mean difference in terms of color?
Well, yeah, or a typical cooking time, because I mean it varies across pulses.
When you compare with chickpea, I can say this is far easy to cook. So
how about with lentils? Lentils seem to be pretty easy to cook.
Yeah, so I think very similar to lentils.
Okay, all right.
So cooking time is.
Okay, so seed shattering and uneven maturity are the main problems for mechanical harvest. How are you approaching that? That's one of the questions.
Yeah, seed shattering was a problem with the land races, but now the latest improved varieties, we have tackled them. And so we have got varieties which do not shatter. But then the maturity, we have got synchronous maturing variety, but what we found is the maturity is highly, there is influenced by the environment. So once mungbean starts spotting, you should not irrigate the crop. So if you start irrigating the crop, there is tendency for new flushes to come. So you need to stop irrigating, and but again, we'll still keep working on it. So that because I know that during the International Congress, there were discussions on this. And now the trend has been to develop early maturing types. But now in the Congress, one of the things which we learned was there was demand for medium maturing varieties also. So that that can be, the yield is important to get the good yield. And in countries like Myanmar, mungbean is harvested at least 3 times. So when we have mechanical harvesting or a synchronous harvesting variety, we have to make sure that the yield levels are comparable. Otherwise, farmers, get into loss.
Right. Okay. Another question is if you have any information on influence or impact of various isolates in specific pathogens for your disease screening trials. So how much diversity is in the pathogen when you do this screening?
Yes, so I think the mungbean yellow mosaic disease is the one which surprised us. And that was the first thing when I joined in this job. I was surprised to see the susceptibility of the current at the time those varieties, resistant varieties to the new strain. So mungbean yellow mosaic is caused by a bigomovirus. It keeps on changing, and that is why it is important that we find alternate sources of resistance. So we keep the competition and we move with the pathogen. So that's definitely the one which has been a challenging one. And the brood kits also, I think, is not with the already I mentioned about the analyst one. So we have to make sure that when we screen, we make sure that thorough screening is done with the viruses or the insect biotypes.
Okay. Question here is from Nigeria is what is your team doing in popularizing mungbeans in Nigeria, which they don't seem to be a major crop, but seem to have good potential?
Thank you. We did popularize mungbean 4 years ago as part of a UK aid project, which again, through the network, we developed lines, we shared with one of the partners. There was a presentation in one of the conferences, and I have to follow up regarding whether any variety has been released. There was very good performance, couple of lines were early maturing, and so I hope they are in the release process. But thank you for that question. I will follow up with our colleagues in Nigeria.
Okay. How attractive it is for youth entrepreneurs, mungbeans in Africa? And what strategies can be developed to attract youth into this type of entrepreneurship?
So yes, thank you. A very nice question. So in East Africa, already in Kenya, Tanzania, and Uganda, and in Ethiopia and Mozambique, these countries already mungbean is being grown, but particularly in Kenya and Tanzania. And very good, the varieties are there, and it is a good progress has been made. And there are private companies. There was a company, one of the companies attended the mungbean conference in Thailand, which I was very happy. And so I need to, mention that to address this issue, we have got worldwide as a consortium called the Africa Vegetable Breeding Consortium. So there is a Asia one, we call as APSA, in which all the vegetable crops are there. But in the Africa Vegetable Breeding Consortium, mungbean is a crop. Is being included, which is for me a very welcome one, because that is from the feedback we got from our stakeholders. So as part of that, every year we showcase our new improved lines, either in East Africa or in West Africa. So last year it was in Arusha. So this year we will have a field day showcasing the new lines, which will be available in advance to the private sector, so that they get leverage of, in advance, 2 years in advance before it is released to the public. So there is good, interest. And there was this Dr. Osi away from Senegal. She works at the Virginia Tech University. She was there promoting in the conference held last month, promoting Mungbeans in Senegal. And so there has been very good interest in West Africa too. And I think there is great potential, especially as a crop which is for addressing the malnutrition, because of studies have been done, which is that there is good acceptability of the taste, and the kids, they love them.
I cannot help but asking this question. I noticed that at least in East Africa, cowpea leaves are consumed. Would there be a potential for mung bean leaves to be also included in the diet?
In West Africa, I know cowpea leaves are being consumed. So during the last meeting in Arusha, couple of the private sector colleagues from West Africa, they said that mung bean leaves are also consumed, but very rarely. But tender pods of mung bean is consumed. The mung bean leaves, when we did analysis, it's very high in iron content. So tender leaves are being consumed, but not very popular. But it's tender pods are consumed, and also of course the grains.
Okay. Yeah, is there any resource in terms of recipes? Because there's a question here from Ethiopia that mung beans have potential, but people may not know how to consume it. Is there any resource that you can indicate to people? I took the liberty to share your email address for people to contact you directly, and I'm sorry if they're gonna flood your email.
No, thank you, Carlos. That is why in my presentation I had one slide on recipe, because it is not that difficult to develop varieties, I can say, as a breeder. But the important thing is to make sure that the variety is accepted by the farmers, and you have to, and if it is a crop is new, you make sure that recipes are given. That is why when in the African Union project in Uganda, there were very good recipes in Kenya. There are lots of good recipes in Asia, but we need to make sure it is customized for the community in that particular country. So I will be happy to share. There is a book on mung bean recipes we developed for Uganda. I'm sure it can be easily adapted or customized for Ethiopia.
Okay, if you can share that resource with me, I can share it with the network later, or the person that asked a question reach out to you directly. We have time for one more question here. Why some seeds coming from the same seed source upon cooking they don't cook well? Is that because of the physiological stage that they have been harvested? Why is there variation within the same batch in terms of softness and cooking time?
Yes, so I think those improved varieties, this should not be an issue. But then, so there could be instances if, especially in terms of if the crop has been subjected to stress, especially heat stress or any other stress, then there could be, these issues could come up. So, but the improved varieties, I'm confident that we have, like those the varieties do not have this problem. But that is why it is good to have varieties which are suited to or adapted to these under stressful conditions. So if there is subjected to heat stress, these issues could come.
All right. Dr. Nair, Ram, thank you so much for an amazing presentation. A lot of questions. As I said, the audience has your email if somebody wants to reach out to you with further questions. Thank you so much for your presentation. Next week, we have the last seminar of this series by Dr. Gustavo Rodriguez from Universidad Nacional de Rosario in Argentina, talking about exotic genes to improve tomatoes. Please join us at the same time. Appreciate your participation. I hope everybody has a good weekend and a good rest of the day. Thank you so much, Ram, and have a great day.
Thank you very much, Carlos. Thanks everyone for listening, and we'll be happy to answer any questions. Please reach out. Thank you for your attention again. Thank you again, Carlos, for this opportunity.
Thank you, Ram.
Much appreciated.
Thank you.
Thank you.
Bye-bye.